going to get set up here. Thank you very much, Tom, for inviting me to share this grand rounds with you and for introducing the topic of EGF receptor mutant lung cancer to everybody. So what I'd like to do today is you can't hear?
You want to use your hand?
Is this better?
Yeah.
It is better. Okay, I just have to talk. I'll figure it out. So, okay. So today what I'm going to do is I'm going to tell you a little bit about the work that we're doing to address the issue of resistance in EGF receptor mutant lung cancer and overcome some of the major challenges that are still present, as Tom told you about. So acquired resistance to EGF receptor inhibitors occurs in virtually all patients with EGF receptor mutant lung cancer, usually on average within a year of treatment with these agents. Acquired resistance is mostly due, in most cases, to a secondary mutation in EGF receptor, the T790M mutation. Other mechanisms of resistance include activation of bypass signaling pathways, like amplification of the MET receptor tyrosine kinase, or amplification of HER2.
This might be easier.
I'll just try this. Is it on? Is this?
Yeah.
This is much better.
Yes.
I can hear that it's much better. Okay. So one can also find mutations in downstream signaling pathways like PI3 kinase mutations or occasionally BRAF mutations in drug-resistant EGF receptor mutant lung cancers, and phenotypic changes in tumors, such as epithelial to mesenchymal transition and the transformation of the tumors to small cell lung cancer from lung adenocarcinomas. So when we think of resistance, what we want to be able to do in patients with EGF receptor mutant lung cancer is we'd like to be able to first find strategies to overcome resistance when they do develop resistance. Even better, ultimately, what we'd like to be able to do is to improve therapeutic strategies up front so that we can delay or even, hopefully one day, prevent the emergence of resistance. However, to achieve these goals, there are several major challenges. And these are, first of all, that as we develop new drugs and new targeted agents, patients are likely to undergo treatment with multiple different drugs over time. And we really don't know what happens to the tumor and how the tumor evolves if you treat them sequentially with a targeted therapy, combinations of targeted agents and immunotherapies. And so here we've just coined the term extreme resistance. And what we'd like to do is we really need to understand and figure out ways of overcoming extreme resistance to be able to really impact this disease. Not only that, we also have to decide how to best sequence therapies. With all of the new targeted therapies to EGF receptor that are being developed in combination strategies, people are really trying to figure out whether using some of these therapies upfront is going to be beneficial compared to using them sequentially. So do you treat a patient with erlotinib and then treat them with afatinib plus cetuximab and then with AZD9291? Or do you take AZD9291 all the way up front? What is going to be better and give the best impact on survival for patients? We also have to identify new vulnerabilities of resistant cancer. So we know that resistant tumors that have the T790M mutation are still sensitive in many cases to drugs that block the activity of mutant EGF receptor. But there are many other mechanisms of resistance. We don't fully understand the molecular changes that underlie many of these changes, and we don't know how to target that. So we really still have a lot of work to do to understand the vulnerabilities of these resistant cancers. And then we really have to figure out how to counteract the plasticity of signaling pathway alterations that can occur in cells. And what I mean by this is signaling plasticity is, I think you've all probably heard of adaptive resistance or of feedback pathways and feedback activation, so that when you effectively block one pathway, you're often turning on other signaling pathways. And so how do we deal with that? Because that can also lead to resistance. So these are really some of the major challenges that we face in the field in terms of thinking about tackling resistance. And so what I'd like to do today is I'd like to tell you about some of the experiments that we're doing to address really these different various points. So first of all, how do we study drug resistance and how do we study these problems? We use various different model systems. We use mouse models, both genetically engineered mouse models and xenograft models. We use drug-resistant cell lines, and then we also use specimens from patients, and in particular specimens from patients who've developed resistance to different EGF receptor inhibitors. Several years ago, we developed genetically engineered mouse models in which we can turn on expression of mutant EGF receptor, many of the different mutants that we'll talk about today, specifically in the lung epithelium. And these mice develop lung adenocarcinomas, as you can see here in these MRIs, and when we treat them with erlotinib, the tumors go away. So we can really follow the evolution of a tumor using magnetic resonance images in these mouse models and using drugs that are used in patients with EGF receptor mutant lung cancer. Not only that, we can actually take these mice and we can treat them long-term with some of these tyrosine kinase inhibitors. We can see the initial responses and we can see drug-resistant tumors emerge as well. So we can model the type of tumor, we can model the response to therapy, and we can model drug resistance. And when we look at the resistant tumors, we see that many of these resistant tumors have changes that have been found in patients with EGF receptor mutant lung cancer who've developed resistance to tyrosine kinase inhibitors. So in addition to the mouse models, one of the things that we've been doing here at Yale for the past couple of years is working with Scott and Gettinger and Anna Wirtz has really been to set up a ReBuyOps program. And what we do is when a patient develops resistance to a targeted therapy and has a thoracic malignancy, we enroll the patient on the study. And so far, we have enrolled over 100 patients on this study, many of whom have EGF receptor mutant lung cancer, some who have ALK-rearranged lung cancer, and others who are enrolled on other clinical trials of targeted agents who don't have an obvious known driver mutation in the lung cancer. But what we can do with this is we can take the resistant tumor and we can compare it to the tumor before treatment. So we can really see what the molecular changes are that are happening during treatment in that patient's sample. And we can do this by evaluating the histology of the tumors, working with some of the assays that have been set up in the tumor profiling and molecular profiling lab led by Jeff Sklar, Zenta Walter, and Mimi Wang. And then we can also do studies using targeted sequencing, whole exome sequencing approach to really discover new changes that happen in these resistant tumors. And so we can interface the work that we do with our genetically engineered mouse models, our cell lines, with this work that we do in patient samples in our studies. So first of all, what I'd like to tell you about is some work that we've been doing to really understand resistance to sequential treatment with EGF receptor inhibitors. And this is work that has emerged, came from, was done mainly by Valentina Pirazzoli, who was a postdoc in the lab. And it started out from the data that we had on afatinib and cetuximab that Tom introduced previously. So now, if a patient has T7, is found to have a T790M mutation in their tumor, there are options for trials with third-generation tyrosine kinase inhibitors like AZD9291 and Clovis 16886. They can also, there has been a trial of afatinib plus cetuximab, which was just published recently, showing about a 30% response rate in this patient population to this drug combination. And this drug combination we found several years ago together with William Pao when we were at Memorial Sloan Kettering. And if we took mice with the L858R plus the T790M mutation and we treated them with afatinib alone, which was the irreversible tyrosine kinase inhibitor, the mice didn't respond to the drugs. If we treated them with cetuximab alone, they didn't respond to the drugs. But what we saw is that they had really incredible responses when they were treated with a drug combination. And we think that this is due to the fact that when you have cetuximab there, you also get downregulation and degradation of EGF receptor in addition to inhibition of phosphorylation of the receptor. And so that really brings the levels of mutant EGF receptor below a threshold. So when this was tested in patients, as Tom showed you, there's about a 30% response rate. But there's the median progression-free survival is 4.7 months. So what that's telling you is that patients are still developing resistance to this drug combination too. And we were thinking, what is the mechanism of resistance to this drug combination? So what Valentina did is she took mice that had L858R plus T790M tumors, and she treated them with afatinib plus cetuximab until they developed resistance to that drug combination. And so, and then what she did is they developed resistance, and she took the tumors, and one of the things that she noticed right away was that the tumors were histologically very different from the tumors before treatment. They had, they were much more poorly organized and poorly differentiated. They had markers of adenocarcinoma still, but they had significantly reduced levels of E-cadherin, for example. And they had the structure, the pattern of vimentin expression was completely changed. So the tumor was much more poorly differentiated when it was resistant. And the other thing that she observed was that in these resistant tumors, although she had inhibition of EGFR and HER2, as predicted by the afatinib plus cetuximab combination, she saw that all of these tumors had higher levels of phospho-S6, mTOR signaling. And so what she did is then she took mice who had resistance to afatinib plus cetuximab, and she treated them with an mTOR inhibitor. And what she saw is that the tumors responded dramatically to addition of an mTOR inhibitor to the afatinib plus cetuximab combination. Now, we did these experiments. We had Xenograft models as well. So we put together sort of these Xenograft models, the genetically engineered mouse models, and made this observation. But one of the things that we were really interested in seeing was whether this was something that could be applicable to patients. And so this is where we went back to our ReBuyOps study, and we looked in our cohort of samples from patients with EGF receptor mutant lung cancer, and we did have some who had developed resistance to afatinib plus cetuximab. And so what we were able to do there was we sent some samples to Foundation Medicine, where they did targeted sequencing of exons of about 200 genes. And one of the things that they found was in one case, and this was a case from Vanderbilt University that through William Pao and David Carbone, was that there was a patient who had mutations, who had acquired mutations in NF2, inactivating mutations in NF2. And NF2 can lead to, loss of NF2 can lead to activation of the mTOR signaling pathway. And in fact, in cell lines, when we knock down NF2, this desensitizes the cells to treatment with EGF receptor inhibitors. We had another patient who had a TSC1 mutation. This is a patient from Yale who had a mutation, an inactivating mutation, and loss of the other allele of TSC1. So TSC1 is part of the tuberous sclerosis complex. TSC1, TSC2 regulate mTOR signaling. And so in this patient, similarly, if you knock down TSC1, you desensitize cells to treatment with an mTOR inhibitor. So this gave us an indication that there might be situations in which you have genetic events that occur when you acquire resistance to afatinib plus cetuximab that lead to hyperactivation of mTOR signaling. Now, the mTOR pathway can get activated in many, many ways, but it still seems to be a therapeutic target for intervention in this type of situation. Whether mTOR pathway activation is important for resistance to some of the third generation tyrosine kinase inhibitors, we don't know, and of course it's something that we're very interested in and actively investigating. So, to summarize this, we did experiments in many of our preclinical models, and what we think is happening is that if you have a TKI-resistant EGF receptor mutant tumor that is still sensitive to afatinib plus cetuximab, you treat it, you get a response, eventually you get resistance, and that this resistance is due to mTOR activation, at least in some cases, and that when you use then, you combine this with an mTOR inhibitor on top of the EGF receptor inhibitors, you can resensitize the tumors and get responses. Of course, ideally, you don't want to use this triple drug combination in patients, but this allows us to think ahead with some of the newer third generation inhibitors, whether it would be worth using them in combination with some of the new mTOR inhibitors as well going forward. So I just wanted to stay on the topic of afatinib and cetuximab and tell you about how we're doing experiments to really try to understand also how to best sequence therapies. So we have this combination that can overcome resistance to EGF receptor TKIs. And as Tom talked about in his talk, what happens when we use this combination up front? And so there's a clinical trial that is going to happen through the Southwest Oncology Group and that Sarah's going to lead on the clinical end. One of the things that we can help with from the lab is we can try to see whether there's preclinical rationale for doing these types of studies. And so what we did is we took mice with EGF receptor mutant lung cancer, L858R mice, so not the T790M. These have never been treated, don't have resistance, and we treated them either with erlotinib, with afatinib, or with afatinib plus cetuximab, long term. And so one of the things that we saw is that if you treat the mice with erlotinib or with afatinib, they develop resistance, and they develop resistance within a time frame that we'd previously shown in other studies. What we noticed is the mice that are treated with afatinib plus cetuximab, only 50% of these mice develop resistance, and it takes them double the amount of time to develop resistance. And so this really is promising as we think about moving this therapeutic strategy up front in patients with EGF receptor mutant lung cancer, and that's hopefully what's going to happen with this clinical trial as it moves forward. So I have two other brief vignettes that I wanted to share with you, and those are about some of our strategies that we're using to think about how we can identify new vulnerabilities of resistant cancers and how we can counteract signaling plasticity. So first of all, we've talked a lot about T790M mutant, EGF receptor mutant lung cancer, but what about all the rest? And so I'm not going to tell you about this work today, but I just wanted to point out that we have used some of our mouse models and interface those with studies of human samples to really establish a role for HER2 amplification and for reduced expression of NF1 in EGF receptor mutant lung cancer in TKI-resistant disease. So where are we? What are we doing now? What are the types of things that we're thinking about? And so once again, one of the things that we've been interested in doing is really going back to our rebiopsy protocol and taking biopsies of patients who developed resistance to an EGF receptor tyrosine kinase inhibitor, either erlotinib or gefitinib or afatinib, compared to the pretreatment specimen, and performing whole exome sequencing of these pretreatment and post-treatment tumors to look at what changes are occurring at the DNA level in these tumors. And so we're in the process of analyzing these data right now, but what I wanted to show you is a snapshot of the frequency of number of resistance mutations that you find in these samples. And you can see that you can go from 0 acquired mutations to about 100 acquired mutations per sample. And this spans over all of the different mechanisms of resistance. So it's not like all the T790M tumors are here and have fewer acquired mutations, and others, and the rest are here with more mutations, as you might expect from a tumor that doesn't have an obvious oncogenic driver. So right now we're in the process of really studying the mutational profiles of these, and then looking at some experiments that we can do to really validate the role for some of these mutations in resistance to hopefully find some new vulnerabilities of the cancers. One of the nice things that we can do from these studies is that not only can we analyze the genetics or the genomics of the tumors, we can also grow the tumors in patient-derived xenografts. So we can take tumors, we can implant them into mice, and this is an example of one of our patient-derived xenografts and work done by Marianne Melnick, a summer student at Penn State, Lam, and Anna Wirtz. And this is, just to show you, this was a liver biopsy from a patient who was treated with erlotinib and had been treated with chemotherapy as well with an exon 19 deletion. didn't have any known mechanisms of resistance. The tumor was implanted into the patient-derived xenograft, and you can see the tumors were treated either with erlotinib, with afatinib plus cetuximab, or no treatment, and you don't really see significant responses. They still have tumors even throughout the course of treatment. And when we go and we look at the signaling pathways in these tumors, we see that the drugs are working. They're inhibiting the phosphorylation of EGF receptor. But what we see in terms of signaling is, for example, here we see that you still have high levels of activation of AKT signal, phosphorylation and signaling, suggesting a potential vulnerability of that tumor to activation of that pathway. That's a pathway that we would expect to decrease if the drugs are functioning properly. And so this is where we can use these types of models now to test new drugs and new drug combinations. So finally, I'd just like to take a couple of minutes and tell you a little bit about some of the work that has been done by Xiaolin Song in the lab, and to really figure out how signaling pathways can change very rapidly in the cells. And some of the, this is, I think, one of the major challenges in dealing with drug resistance, and that is that signaling pathways can change very rapidly, even in vivo. And so Xiaolin was really interested in understanding the role of mutant EGF receptor and how it works with its other heterodimerization partners, either HER2 or HER3. And she especially focused on HER3 because there was evidence that mutant EGF receptor might pair with HER3 to activate the PI3 kinase pathway. And that was an important signaling pathway. And so what she did is she did some work very recently in which she took mice that have EGF receptor mutant tumors in which she could inactivate, get rid of HER3 in established tumors using some genetic tricks. And so when she did this, what she saw, and you can pay attention to this graph right here, and look at the red line, is that when she gets rid of ERB3, the curve kind of flattens out. It sort of stabilizes disease, but then it's starting to grow after a little bit again. The tumors are picking up and growing again. And when she did Western blots on these tumors, she was really surprised. We were all very surprised to see that when she got rid of ERB3, as you can see here, the levels of phosphorylation of EGF receptor and of adapter proteins and of proteins that directly link to EGF receptor were higher than in the tumors in which ERB3 was present, suggesting that you get a shift in the heterodimerization partners that are present when you get rid of one of the pathways. So what does this mean? This means that the tumor recovered really, really fast. You got rid of a molecule, and then it figured out a way of getting around it really quickly. Now, what does this mean therapeutically? This means that potentially you can change the vulnerability of a tumor cell by altering these different molecular pathways and these heterodimerization partners. And in fact, what Xiaolin has seen is that if she takes tumors induced by the EGFR L858R plus T790M mutant, and she gets rid of ERB3 and then treats them with cetuximab, these tumors stabilize and don't grow as well. They'll pick up eventually because they'll adapt again, but whilst the other ones that have ERB3 don't. So the other ones that don't have ERB3 are less vulnerable to cetuximab than the ones in which you've gotten rid of it. So the sensitivity of the tumor has changed. So I just want to conclude by going over some of the things that I've told you about today. How we can use some of our preclinical models to find strategies to overcome resistance by finding new mechanisms, by testing new drugs and new drug combinations, and also how we can think about improving therapeutic strategies to delay resistance by looking at combinations up front, eventually by targeting residual cells, which I didn't talk about. And of course, one of the things that I didn't talk about at all in this presentation was the immune system and the tumor microenvironment. And I think that's something that we're interested in and that we're working on, and that hopefully we'll be able to tell you about in the future. So finally, I'd like to acknowledge collaborators here at Yale and elsewhere, and funding sources I have some disclosures too. And that's it. I'm happy to take any questions.